in the electron transport chain, sometimes we get the production of reactive oxygen species. Now, these reactive oxygen species are actually detrimental to the cell because they can cause a lot of oxidative stress and they can inactivate enzymes. So we must have a mechanism in place to be able to remove these reactive oxygen species from the cell. So if you take a look over here, we have a depiction of the electron transport chain with complex 1, complex 3, and complex 4. I haven't drawn out complex 2 because we're not interested with complex 2 right now. But we also have this other protein in the cell membrane, and it's called nicotinamide nucleotide transdehydrogenase, and essentially it's going to convert NADH into NAD+, and we'll see why that's important in a minute. So essentially, when NADH gets oxidized at complex number 1, it's going to be passed on to FMN, passed on to iron sulfur clusters, and then passed on to quinone. And this quinone, when it gains 1 electron and it persists for too long with only 1 electron, it exists as a semiquinone. And that semiquinone radical, if it persists for too long, it can react with oxygen. When it reacts with oxygen, it can form this radical oxygen, which is going to cause a lot of oxidative stress within the cell if this reactive radical oxygen persists for too long, especially if it's converted into an OH molecule. So the mechanism we have in place to get rid of this reactive oxygen and its OH group is essentially utilizing the enzyme superoxide dimutase. Superoxide dimutase will convert O2 into H2O2. Now, H2O2 will then further be converted into H2O via the enzyme glutathione peroxidase. Where do we get the electrons for this reaction over here? Well, we have reduced glutathione over here. So essentially we use 2 GSH molecules. So we have 2 hydrogens, which are going to be transferred over here, so we get H2O. This is catalyzed by the enzyme known as glutathione reductase. And as a result, we get the product of oxidized glutathione, so GSSG. How do we regenerate reduced GSH just in case we have another oxygen to get rid of? We're going to regenerate this molecule by picking up electrons from NADPH. So GSSH is going to pick up electrons from NADPH, and as a result, NADPH is going to get oxidized into NADP+. When NADP+ becomes oxidized, it can become reduced once again because it can pick up electrons from NADH. This is where we see the role of nicotinamide nucleotide transdehydrogenase. It's going to oxidize an NADH, and it's going to produce NAD+, and those electrons are going to be passed on to NADP+ to yield NADPH. As a result, everything in this process is being regenerated. We regenerate this molecule, we regenerate these molecules, all because we want to get rid of this radical oxygen. Now, this reduced glutathione actually has another purpose. Now, when we talk about enzymes, enzymes are made up of proteins. Proteins are made up of amino acids. One particular amino acid is cysteine, and cysteine has a sulfur group. During oxidative stress, enzymes become inactivated. When they become inactivated, it's because these sulfurs have formed a disulfide bridge. Now, these disulfide bridges are due to a oxidative stress. So we can see over here, this enzyme was reduced because it had these hydrogens, but then it becomes oxidized and we see this bond form, and this bond is referred to as a disulfide bridge. So the enzyme is inactive. We want to get it back to the active state. That's where we see this reduced glutathione come in again. So via the enzyme protein thiol reduction enzyme, we see that the electrons from 2-GSH are passed on to this inactive enzyme, which results back into the reduction of the enzyme and it's active once again. So we can see actually that oxidative stress can cause damage to a cell by inactivating enzymes. So we need this process to reactivate our enzymes. So an early indicator of oxidative stress is inactivation of enzymes. If a lot of our enzymes are inactive, we know that there may be some oxidative stress involved. And that is essentially the pathway in which reactive oxygen species are produced. So they are produced because of this quinone, this semi-radical quinone. And that's essentially how we get rid of these reaction oxygen species.